Hi, I'm Devlina Samantha, a postdoctoral fellow in Professor Chad Merkin's lab at Northwestern University. I would like to begin by thanking Professor Nancy Hsu for inviting me to present in this symposium on nanotechnology, single molecule and single cell imaging in biology and medicine. Today, I'm very excited to be sharing with you some of my recent work on how we can design highly tailorable probes called NanoFlares for analyzing live cells with single cell resolution. Now, if we were to look at a cell through a chemist's perspective, we see that a cell has hundreds and thousands of different molecules which are interacting with each other and participating in chemical reactions, which is what we collectively call life. From an analytical chemistry standpoint, there are many very well-established techniques for studying different classes of analytes in cells. For example, we can study proteins using genetically encoded fluorescent sensors, and we can study nucleic acids using techniques such as FISH and PCR. But still, there exists a large fraction of molecules that are difficult to probe in live cells, and these molecules are called the dark matter of cells. So it is important to recognize that studying endogenous molecules in live cells is a grand challenge. So in today's talk, what I will do is I will start by answering the question, why is it important to analyze live cells? And then we will talk about how scientists have done this so far. After that, we will get into what nanoflares are and why they're particularly advantageous for studying live cells. But nanoflares also have some limitations, so we will briefly talk about these limitations and discuss how we can design next generation nanoflares that overcome these limitations. And the way we have accomplished this is by first developing a new signal transduction strategy that has enabled us to make a new probe architecture. Finally, we'll end with a discussion on what we envision can be done in the future using these new probe structures. Now, if we were to take a step back and look at commonly used techniques to study cellular analytes, we see that most methods, ranging from mass spectrometry to PCR to even several microscopy techniques, don't study live cells. Most methods either look at fixed cells, in which we get no dynamic information, or they look at lyse cells, where we lose all spatial information, or they require the use of physical or chemical transfection methods, which are not only toxic to the cells, but can also change the expression level of the analyte that we are trying to measure in the first place, and therefore interfere with the measurement. Now, if we could study live cells, then not only can we learn about fundamental biological processes, which are all dynamic, but we can also learn about how diseases progress with time, and we can potentially detect these diseases earlier. So the question that arises is, can we create a general class of probes for live cell analysis? For example, we want to make a set of probes. We don't know yet what their structure will look like, so it's a black box. But can we make these probes so that they enter cells, bind to target molecules, and result in some form of signal? Turns out, nucleic acids like DNA are very desirable building blocks for making a general class of probes like this. And this is because nucleic acids can bind to both nucleic acid and non-nucleic acid targets. For example, I can make a single strand of DNA and it can recognize complementary sequences through hybridization. On the other hand, nucleic acids can also be generated such that they adopt complex secondary structures and are able to bind to non-nucleic acid targets such as ions, small molecules, or even proteins. These nucleic acid sequences are called aptamers. As an example, here I've shown the structure of an ATP aptamer that is bound to two molecules of ATP. Unfortunately, building probes from linear nucleic acid sequences doesn't really help live cell analysis. And this is for two main reasons. First, the cell membrane makes cellular entry of exogenous DNA very inefficient. And second, linear nucleic acids are rapidly degraded by nucleases. So over the past 25 years or so, The structures of nucleic acid-based probes have been evolving so that we are able to detect analytes in live cells. The field has seen a marked shift from simple linear structures, such as those used for in situ hybridization, which was developed in the 1960s, to more sophisticated nanoarchitectures. And the resulting probes have really opened new frontiers in live cell analysis. They have helped not only in the discovery of new biology, but have also been used for bioimaging and disease diagnosis. One such construct, which has been really transformative in the field, is the spherical nucleic acid, or the SNA, developed by the Merkin group. SNAs have highly oriented nucleic acids arranged in a spherical orientation, usually around a metal or organic central core. And this arrangement results in several emergent properties as shown in this table. For example, unlike linear nucleic acids, SNAs are taken up efficiently by cells and are more resistant to nuclease degradation. Based on the SNA architecture, NanoFlares were developed in 2007, which allow live cell genetic and metabolic analyses. Here we start with an SNA with a gold nanoparticle as the core and nucleic acid sequences termed recognition sequences as the shell. Now, the recognition sequence can be anything. For example, it can be a sequence that is complementary to some target RNA, an aptamer that binds to a small molecule, or even a DNA zyme. Anyway, these recognition sequences are hybridized to short fluorophore-labeled strands called flare strands. The gold nanoparticle acts as a quencher. When the target molecule comes in, it binds to the recognition sequence and displaces the flare strand. As the fluorophore gets spatially separated from the gold quencher, a fluorescence turn-on is observed. As an example of how this works, if we were to treat cells using nanoflares that target a particular gene versus nanoflares that do not target anything in the cell, a much higher fluorescence is observed when the cells express the target. This platform has enabled new technologies that were not possible before. For example, detecting cancer metastasis early can really be the difference between life and death. NanoFlares were the first platform that allowed one to detect circulating tumor cells in live form based on gene expression. Because the cells are alive, they can be isolated and cultured further so that we can test their response to experimental therapy. So we can screen drugs that are effective for treating the disease in high throughput, and we can do this in a patient-specific manner, meaning this creates an avenue for personalized medicine. We can also diagnose diseases based on genetic markers by imaging live animals. However, like all fluorophore quencher-based platforms, nanoflares also have limitations. For example, nuclease degradation, dehybridization of the flare strand, or destruction of the gold thiol bond that attaches the DNA to the gold surface all lead to separation of the fluorophore and the quencher, and therefore can give rise to a false positive signal. So the question we asked is, can we develop a strategy that does not rely on a quencher so that it can overcome these limitations? Now, we should note that the propensity for false positives limits quantitative capabilities of nanoflares. Additionally, because the recognition sequence is partially blocked by the flare strand, the rate at which the probe binds to the target also slows down. And of course, an obvious limitation of nanoflares is that there must be a nucleic acid recognition sequence for a given target so that we can detect it. So at this point, it is very clear that we need to develop a quencher-free strategy. How can we do that? To do this, we decided to use a fundamentally new chemical strategy called forced intercalation. So there are these DNA intercalators that are derivatives of thiazole orange, which have internal rotation-dependent fluorescence. So if these dyes are chemically conjugated to single-stranded DNA, upon light excitation, they can undergo internal rotations about their methane bridge, as a result of which they are non-fluorescent. However, when these single-stranded DNA containing the dye are hybridized to complementary sequences, the dyes are forced to intercalate or fit between DNA base pairs, which restricts their rotation and turns on their fluorescence. So the idea that we had was that what if we were to take an aptamer sequence and then use this dye as a base surrogate, such that when the aptamer binds to its targets, the dye would also be forced to intercalate between the different DNA base pairs and as a result fluoresce. So we went ahead and showed that fit aptamers can be designed for the most commonly used aptamer target binding modes in literature. And we did this for targets as simple as metal ions to much more complex targets such as proteins. One example of the metal ion is mercury, which is a toxic heavy metal, and an example of the protein is thrombin, which is involved in the blood clotting process. For the sake of time, I will not go into detailed data, but instead highlight what the main findings of this study are. We observed that fit aptamers result in up to 20-fold fluorescence turn-on upon target binding, and as they don't rely on a quencher, they avoid false positive signals, unlike fluorophore quencher-based systems. Now, FRET-based platforms are also quite popular for avoiding false positives, but what we found is that in certain cases, fit aptamers give higher signal-to-noise ratio compared to FRET-based systems. Another advantage of this platform is that they are kinetically superior to systems that require strand displacement. Earlier I talked about how the recognition sequences in NanoFlares are partially blocked by the flare strands. So what we showed is that when we use a single-stranded fit aptamer, it responds at least 5 times faster compared to when it is partially blocked. And we are also able to detect targets with high sensitivity down to nanomolar levels in even complex media such as human serum. So in a nutshell, fit aptamers are the only light-up aptamer probe that can detect a wide variety of targets with a single modification. Now that we have developed this quencher-free strategy, it positions us to create the next generation of NanoFlares using these fit aptamers as recognition sequences on a nanoparticle core. So we don't really need to use gold anymore because this strategy is quencher-free. So it really provides access to biodegradable and more biocompatible probes. So in this new design, the nucleic acid shell facilitates cellular uptake and can be used to detect analytes using fit aptamers. But excitingly, now we can use a protein core that enables us to make site-specific modifications. For example, we can attach DNA to the lysine residues. Moreover, now we can detect additional analytes using the recognition properties of the protein that we may not be able to detect with DNA alone. Of course, this strategy is going to be resistant to false positive signal because of the use of these fit aptamers and would therefore provide us with greater quantitative capabilities. To test our hypothesis that we can use these protein spherical nucleic acids for live cell analysis, we developed a protein spherical nucleic acid with an I-motif fit aptamer as the nucleic acid shell and beta-galactosidase as the protein core. So in this case, when the pH is dropped from 7.5 to 5.5, the I-motif undergoes a structural transition from a stretched conformation to a quadruplex structure. And when this quadruplex is formed, the intercalator dye, which is thiazole orange in this case, is forced to intercalate between the base pairs of the I-motif quadruplex and result in a fluorescence turn-on. So in buffered conditions, we see that up to 9-fold fluorescence turn-on is observed using an I-motif sequence, whereas if we were to use a control sequence that does not undergo this type of conformational change, we see that the fluorescence remains unchanged. We next wanted to test whether these protein spherical nucleic acids or pro-SNAs are capable of reporting intracellular pH changes. For this, we treated the cells with I-motif containing protein spherical nucleic acids and then clamped their pH at a specified pH using a clamping buffer. Then, using flow cytometry, we analyzed a large population of cells, one cell at a time. When we looked at the fluorescence of the cells that were clamped at a pH of 5.5, we found that these cells were almost twice as fluorescent as cells that were clamped at a pH of 7.5. However, cells that were treated with a control pro-SNA that does not form the eye motif sequence does not show any difference in fluorescence at the two different pH. We next wanted to test the hypothesis that using a functional protein core, we can detect analytes for which there are no known nucleic acid-based recognition sequences. As an example, we chose glucose because of its central importance in biology, and also because there are no known recognition sequences that can bind to glucose with a biologically relevant binding affinity. To detect intracellular glucose levels, we developed a new two-step assay. So it is known that glucose oxidase catalyzes the conversion of glucose to gluconic acid and produces hydrogen peroxide in the process. Therefore, we synthesized glucose oxidase spherical nucleic acids. So in the first step, we incubated the cells with the glucose oxidase SNAs, which resulted in the formation of hydrogen peroxide intracellularly. And this hydrogen peroxide can then be sensed using a commercially available fluorescent derivative dye, which is a boronic ester of fluorescein, and non-fluorescent. Upon reacting with hydrogen peroxide, the boronate groups are cleaved and highly fluorescent fluorescein is formed, which indicates the presence of glucose. This new assay has a really remarkable 120-fold turn-on in buffer in the presence of glucose. And this turn-on is highly selective for glucose over other sugars. And the reason for this really high specificity is because we are using an enzyme for detection. Now in cells, when we treat the cells with the fluorescein derivative dye, we see that the fluorescence increases compared to the untreated cells. Now this is expected because this dye senses hydrogen peroxide and some amount of it is formed naturally in the cells. But when we look at the fluorescence of the cells that are also treated with glucose oxidase SNAs, we see that it's increased by up to 12-fold, indicating that we are able to detect glucose intracellularly. We have validated this assay in 9 different cell lines, representing a mix of different cell types, such as normal cells, cancer cells, and so on. To summarize, NanoFlares represent a highly tailorable class of intracellular probes. By choosing a DNA sequence that can recognize a target, we can detect the target in live cells. Now we have developed a next generation platform based on a protein core that allows us to detect analytes not only using the DNA as the recognition element, but also the protein. This really opens up a realm of possibilities, particularly in terms of multiplex detection. Going forward, using these probes, we can study dynamic processes, which can potentially lead to the discovery of new biology. Because phytaptamers bind to their targets, we can also track analytes and learn what they do in a cell. Using enzymes as cores allows for amplified sensing, which will be useful for detecting low abundance targets and potentially help to diagnose diseases earlier. Importantly, because protein-based techniques are routinely used to analyze cellular molecules, the development of this new platform would allow these proteins to be delivered into live cells and allow making measurements that could not be made before. One of the most important and also challenging directions forward is the ability to absolutely quantify analyte expression levels in cells. With this, we come to the end of my presentation. I would like to thank Professor Merkin for being an amazing advisor and giving me the opportunity of working in his lab. His mentorship and support have been crucial to this work. I'm also grateful that I get to work with a very talented colleague, Sasha Ebrahimi. A big shout out to my other collaborators who have been involved in this project and the Merkin Group as a whole for creating a stimulating and exciting environment to work in. Last but not the least, I'm grateful to the funding that has supported this work, including a postdoctoral fellowship from the International Institute for Nanotechnology and an HHMI Hannah Gray Finalist Award. Thank you for your attention, and I would be happy to answer your questions.